This video will model the process of DNA replication for you. DNA replication starts with the orange molecule, helicase, an enzyme. This unwinds the DNA. Then DNA polymerase, the gray enzymes, come in and extend the new chain of nucleotides. Nucleotides, as you know, have a 3' end and a 5' end. In this video, the 5' end is indicated by the cut out arrow, and the 3' end is indicated by the extended arrow. The projection to the top of this A nucleotide is the nitrogenous base, and it is where it will connect to a complementary nucleotide on the opposite strand. In this case, a T nucleotide. The first thing I need to do is build a strand of DNA out of these nucleotides. In this video, you can see me matching up the 5' and 3' ends of the nucleotides. Once I have one strand, I then create the complementary strand, matching T's to A's and C's to G's. Once my strand of DNA is complete, I will start the process of DNA replication. DNA replication again uses helicase, which is the orange enzyme you see there. So I will feed the DNA into the helicase at the active site, making sure that 5' and 3' ends are facing the right direction. As you can see, the helicase breaks the hydrogen bonds between the nitrogenous bases and pulls them apart, so the DNA, the double strand, is separated. Once a good amount of DNA is separated, DNA polymerase will attach to one of the strands once again lining up the 5' and 3' ends in the correct order in the active site. Then, new nucleotides complementary to those that exist will attach to the strand of DNA at the active site. DNA polymerase will continue moving along the strand of DNA and creating this new complementary strand. Notice that the new strand of DNA is made out of gray nucleotides. This is to signal that it is the new strand, and the colorful strand was the template strand in the original DNA double helix. It is important to note that this process is happening on both strands at the same time. Both of them have a DNA polymerase attached and are creating complementary DNA molecules. If we speed things up, you can see how two new complete DNA double-stranded molecules will be made out of this original template double helix. Notice that the DNA polymerase molecules move in opposite directions on the two different strands, since these two strands are anti-parallel, meaning their 5' and 3' ends are opposite from each other. The DNA polymerase synthesizes the new DNA in the direction of 3' to 5' on the template strand and 5' to 3' on the new strand. Here we have our two new double-stranded DNA molecules. Note that each of them has one strand that is colorful and one strand that is gray. That is why we call DNA replication semi-conservative. Each strand receives one of the template strands, the colorful ones, and creates a new complementary strand, the gray one. This means that part of the original DNA is in each of the new DNA molecules.